everyone, and welcome to Echo My Drop, where we bring you fresh insights into how cutting-edge science is helping us tackle the world's most pressing environmental problems. I am Dean, and I'm thrilled to be kicking off today's episode, where we dive deep into something you might not expect to be environmental heroes: genomics and proteomics. Now, let's take a moment to think about what's happening around us. We are living in a world that's facing environmental challenges like never before. From the small intercities to the microplastic floating in the ocean, and from polluted rivers to degraded farmlands, the signs of environmental stress are all around us. Industrial discharge, deforestation, oil spills, urban expansion, rising temperature, these aren't just headlines. They are real issues as they are changing the way ecosystems function. The scary part? Many of these changes are invisible to the naked eye, especially in the early stages. By the time we notice fish dying in rivers or forests looking dry and lifeless, the damage has often already been done. So, how do we stay one step ahead? How do we catch these problems before they become disasters? That's where science gets really interesting. Traditional environmental monitoring is like checking someone's health using only a thermometer. Sure, you might catch a fever, but you will miss so much else like inflammation, stress hormones, or early signs of chronic disease. Now, apply the idea to the environment. We've been relying on basic physical and chemical indicators like pH levels, temperature, and visible pollutants. But these methods cannot always pick up on the biological response happening inside organisms when they face pollution or climate stress. They don't tell us how an organism is coping or whether it is already on the brink of collapse. This is where functional genomics and proteomics come in. These fields allow us to study genes and proteins of organisms, giving us a molecular level clues about health, resilience, and adaptation. Let me break it down a bit. Functional genomics looks at which genes are turned on or off in response to different environmental conditions. For example, when a plant is exposed to a heavy metals in the soil, it might activate genes related to detoxification or stress resistance. This powerful info because we are seeing how life itself adapts or struggles to survive under pressure. Now, proteomics adds another layer. Genes are like instructions, but proteins are the actual doers. They carry out the tasks, breaking down toxins, repairing damage, signaling distress. Proteomics help us see what's really happening in real time inside the cells of an organism. Together, these tools act like a high-tech stethoscope for nature. They let us listening on how organisms respond to environmental threats, not just reactively but proactively. And don't worry, we are not going to throw a textbook at you. Throughout the podcast, we are going to bring you real stories. From microbes cleaning out oil spills to how proteomics is helping us detect pollution in fish before it's visible to the eye. We'll also touch on future possibilities, like using environmental DNA to track water quality, or genetically modifying microbes to work smarter in bioremediation. So, whether you are a science student, an environmentalist, or someone who just wants to understand what's happening behind the scenes in nature, this episode is for you. Because let's face it, if we want to build a more sustainable future, we need to go deeper. And that means understanding ecosystem at the most fundamental level, at the level of genes, proteins, and communities of microscopic life. So get comfy, grab your favorite drink, and let's explore how tiny molecules can lead to massive environmental solutions. Coming out next, Ernest will introduce us to the tiny but mighty microbes that are transforming microenvironmental clean out through the power of metagenomics. You won't want to miss it.
Thanks, Tim. That was such a powerful introduction. Now, let's take things even deeper. Hi, everyone. I am Agnes, and I'll be your guide through one of the most fascinating part of the environmental science, the invisible world of microbes, and the powerful tool we use to understand them, metagenomics. Let me start with a simple but mind-blowing fact. In a single gram of soil, there can be over a million of microorganisms. That's more living being in your palm than there are people on Earth. And yet, for the longest time, scientists could only study a tiny fraction of them. Those we could grow in a lab. That's like trying to understand an entire city by only interviewing 5 people. That changed with the rise of metagenomics, a technique that allows us to extract and sequence DNA directly from the environmental sample. Soil, river water, ocean sludge, even air. It's like collecting the environmental fingerprinting of all the microbes in a sample and then using a powerful software to piece together who they, what genes they have, and what role they play together in the ecosystem. But what makes microbes such rock star in the environmental science? It turns out some of them have superpowers. They can actually break down the pollutants that are toxic to other forms of life. This is the basis of bioremediation, where we use microbes to clean up contaminated environment. Imagine bacteria that can digest oil, pesticide, or even radioactive materials. They don't just survive, they thrive on the mess we make. One of the most famous cases was after the Exxon Valdez oil spill in Alaska. While humans scrambled to contain the damage, microbes begin quickly digesting the hydrocarbon. Since then, scientists have looked for the ways to identify and even enhance these cleanup specialists. Here where metagenomics shine. It allows us to scan polluted environment like oil-contaminated soil or industrial waste site and find out which microbial community are present, what genes they carry, and whether they are capable of degrading pollutants. Let's take a specific example. In soil contaminated with PCBs, a group of toxic man-made chemicals, researchers use metagenomics to identify bacteria that could metabolize biphenyl, a PCB-like compound, as their food source. That's like turning poison into a buffet. They even pinpointed the gene cluster responsible for disability, such as the BPH-ABCDEFG cluster. Another example is bioaugmentation, where we intentionally introduce helpful microbes into the polluted environment to speed up the cleanup. But it's not just about tossing bacteria into the soil and hoping for the best. Thanks to metagenomics, we can track how these introduced microbes behave, how well they survive, and whether they are making a difference. We can even monitor changes in gene expression during the process. And this technology is not just about pollution cleanup. It's being used to protect public health too. Take environmental DNA or eDNA for example. Scientists in New Zealand use high-throughput sequencing of microbial DNA from drinking water and compare it to DNA from different animal feces. By matching the DNA profile, they were able to trace the exact source of feces contamination in river. That's a game changer for preventing waterborne disease. Another cool use of eDNA, biodiversity monitoring. Without even catching or tagging a species, scientists can detect its presence just from the DNA it leaves behind. That means we can monitor fish population, invasive species, or endangered wildlife, all without disturbing their habitat. Metagenomics also helps in agriculture sustainability. Soil microbes play a big role in supporting plant health and nutrient cycling. By analyzing the soil microbiome, farmers and researchers can better manage the crop, reduce the need for chemical fertilizer, and promote healthier soil. So, to wrap it up, metagenomics is helping us turn the invisible into the understandable. It's revealing the vast microbial universe in our environment and helping us harness its power for good. From cleaning up oil spill to tracing the water contamination, from boosting agriculture to protect public health, this technology is transforming how we interact with nature. And guess what? We only scratched the surface. There's still so much to learn about this microbial community, their genes, and their potential to restore our planet. Coming up next, Pauline will walk us through another exciting layer, how the protein these microbes and other organisms produce can tell us even more about how the environment is doing and what we need to pay attention to.
We have all heard of genes. They are like the instruction manual for life, storing the blueprints that tell cells what to do. But here is the thing. Having a manual does not mean much unless someone reads it and acts on it. That is where proteins come in. Proteins are the real deals. They are tiny biological machines that carry out almost every task required for life. They build cell structures. They carry messages. They break down harmful substances. They fight off infections. They provide energy. Without proteins, the instruction in our DNA would just sit there, unused. In the human body, proteins are involved in everything from muscle movement to digestion to brain function. But it is not just in people. Proteins are just as important in plants, animals, bacteria, and even in the smallest microbes in the environment. They are essential for every living thing. And when an organism is exposed to pollution or environmental stress, its proteins are often the first to respond. Now imagine a fish swimming through polluted water. On the surface, it might look fine, but inside its body is already reacting. It begins producing special proteins called biomarkers. These include heat shock protein, which help repair stress damage detoxification enzyme that break down pollutants, and oxidative stress proteins that fight off harmful molecules. These proteins appear long before you see any signs of sickness or death. They are early warning signals that the environment is under pressure. This is where a scientific tool called proteomics becomes very important. Proteomics is the study of all the proteins in a cell or organism. It helps scientists discover which proteins are being made, how much of each is present, and what roles they are playing. You can think of proteomics as opening a toolbox inside the organism. By looking inside, scientists can see which tools or proteins the organism is using to deal with its environment. A healthy organism will show one pattern of protein activity. A stressed one will show another. Proteomics gives us a detailed view of what is happening inside a living system. Traditional environmental monitoring looks at things like pH, chemical levels, or temperature. These methods are useful, but they might miss the early biological effect of pollution. Proteomics tells a different story. If an animal or plant start producing stress proteins, even when water quality seems normal, we know something is wrong. That allows action to be taken early before serious damage happens. It is like noticing someone has a fever before they show other symptoms of illness. Proteomics is already being used in many different environments. In coral reefs, scientists have detected stress protein before coral bleaching occurs. In forests, it helps track how soil organisms recover after deforestation. In polluted wetlands, researchers use proteomics to see how antibiotics from fish farms affect bacteria. Some microbes start making proteins that resist or break down antibiotics. Others stop functioning completely. That tell us exactly how pollution is changing life in the ecosystem in real time. Today, the environment faces many trends at once. Pollution, habitat loss, and climate change are happening together. By the time we see visible damage in an ecosystem, it is often too late. That is why proteomics matters. It gives us a window into how life is reacting at the molecular level. It lets us see stress signals inside organisms early. This means scientists, conservationists, and environmental managers can act faster and more accurately. Proteomics turns invisible stress into visible data, and that helps us protect life more effectively. Proteomics is not just about identifying stress. It is also being used to support recovery and restoration efforts. By seeing how organisms respond to cleanup and conservation, we can measure what is working and what still needs to be improved.
Thanks, Ferlin. There was such a vivid journey into the world of proteins, our cellular messengers and defenders. Hello, listeners. I'm Shell, and now we are going to explore the fascinating worlds of biomarkers and environmental DNA, or eDNA. Think of this as natural molecular warning signs that help us detect, monitor, and even predict environmental issues before they spiral out of control. Let's begin with biomarkers. Imagine if a river could send you a signal saying, "Hey, I'm stressed," or a forest whispering, "There's something toxic in the soil." Biomarkers are how living organisms do communicate with us at the molecular level. A biomarker can be a DNA sequence, a protein, an enzyme, or even a small molecule that appears when an organism is exposed to a pollutant. Think of it like a smoke alarm going off. Not because you can see the fire, but because your environment knows it's there. For example, in aquatic environment like lakes and wetlands, certain bacterial genes become more active in the presence of antibiotics or chemical runoff. Scientists can measure those genes to determine if the ecosystem is under threat even if it looks fine to the naked eye. In one study, researchers investigated a wetland that had been affected by massive fish farming. At first glance, the area looked normal, but metagenomic analysis told a different story. Used biomarkers, they discovered that dangerous bacteria like fusobacteria and elosin microbia were dominating the microbial community. This raised a red flag about antibiotic resistance and poor water quality. They used a tool called leipzig analysis, which helped pinpoint the exact bacteria acting as the biomarkers. What's amazing is they could map this microbial hotspot using Gibbs mapping, like creating a heat map of bacterial pollution. Now, let's shift to the environmental DNA or eDNA. Imagine dipping a test tube into a stream and learning everything that swam into it in the last 48 hours. That's the magic of eDNA. Organisms constantly shed DNA into the environment through skin cells, waste, mucus, even saliva, by collecting samples of water, soil, or air and sequencing the DNA fragment inside. Scientists can detect the presence of species without even seeing them. No nets, no trap, no microscope, just clean, powerful data. And this isn't just about counting fish or tracking frogs. eDNA is being used to monitor endangered species, invasive species, and even pathogens in drinking water. In one case from New Zealand, scientists used eDNA to trace vessel contamination in drinking water. They collect samples from water source and compare the DNA to non-fisher samples from humans and animals. What they found were very specific microbial fingerprints, Escherichia and Enterococcus, which are the classic indicator of waterborne disease risk, that allowed health officials to act before an outbreak occurs. This technology is especially valuable in the world where environmental stress is often invisible until it's too late. By identifying biomarkers and tracking eDNA, we gain a much more sensitive, real-time understanding of ecosystem health. But there's more. Biomarkers can even tell us which parts of an ecosystem are recovering or which species are thriving in response to conservation efforts. For example, if a polluted wetland is restored and we start detecting protein or DNA associated with healthy soil bacteria, there's molecular proof that our strategies are working. This has big implications for policy and management. Imagine proving a river is recovering after cleanup or catching pollution early. That's the power of biomarkers and eDNA in monitoring nature. But we must be careful. These tools require high-quality sampling, advanced equipment, and robust bioinformatics, and we must handle the data ethically, particularly for biodiversity. So to wrap it up, biomarkers and environmental DNA give us an unprecedented window into the invisible world of environmental health. They allow us to detect change early, respond quickly, and track recovery over time. Whether it's a stream, a wetland, or a coral reef, These two are like having a stethoscope to listen to the heartbeat of nature. Coming up next, our speaker 5, Jiahui, will take us to the finish line exploring how these omics tools, genomics, proteomics, metagenomics, and more are shaping the future of the environmental conservation and sustainability.
Thanks, Shell. That was such a fascinating deep dive into the molecular whispers of nature, how biomarkers and environmental DNA are helping us become better listeners and stewards of the environment. Hello everyone, I'm Jiahui, and to wrap up our podcast today, I'm going to take you on a quick journey into the future. A future where functional genomics and proteomics are not just research tools, but key players in how we restore, protect, and coexist with nature more sustainably. Let's start with a powerful thought. What if we could predict environmental disasters before they happen? Not after a fish dies off or a crop failure, but before the first signs of trouble. That's the direction we're heading in with omic-based technologies. By integrating genomics, proteomics, and metagenomics, scientists can model how ecosystems function and how they respond to stress. Imagine a system that constantly monitors soil health in a farm, flagging when microbial balance is tipping toward degradation, or a coastal monitoring station that picks up protein biomarkers of coral stress weeks before bleaching becomes visible. These are not science fiction, they are fast becoming reality. To get there, we need integration and innovation. One of the biggest trends is the development of multi-omics platforms for genomics, transcriptomics, proteomics, and metabolomics data give scientists a detailed picture of what's going wrong, why, and how to fix the issue in nature. Let's look at conservation. As climate change accelerates, conservationists are turning to omics tools to make decisions. For instance, in land restoration, metagenomic profiling helps identify designer microbes that restore nutrients, fix nitrogen, and detoxify pollutants. And what about urban areas? Urban ecosystems face their own stress, like pollution and heat. By studying urban microbiomes, scientists can create greener infrastructure and build more resilient cities. But with all this innovation, we also face real challenges. First, there's the issue of data overload. Omics generate massive data, and making sense of it requires powerful bioinformatics and skill analysis. Second is standardization. For data to be shared and compared globally, we need consistent sampling protocols, quality controls, and data formats. Thirdly, ethics and biosafety. As we develop genetically modified microbes or apply omics tools to sensitive ecosystems, we have a responsibility to ensure that we don't cause unintended harm. Still, the benefits far outweigh the risks. Government, industry, and climate researchers are embracing omics for better monitoring and greener solutions. So, what does all this mean for us? It means that we enter an era before our relationship with nature become more informed, more respectful, and more scientifically empowered. Omics remind us that everything is connected. Every gene, every protein, every molecule tells a story, and it's up to us to listen. That brings us to the end of our podcast, exploring the roles of functional genomics and proteomics in ecosystem health, bioremediation, and environmental conservation. We hope you enjoy this journey through the molecular lens of environmental science. Thanks for tuning in, and remember, science is not just happening in labs, it's happening all around us. in every leaf, every drop of water, and every breath of air.